Hello, a very warm welcome to everyone here and those who are joining us via Zoom webinar. Thank you for joining us for this Life Sciences Masterclass. Now, before we begin, there are some housekeeping announcements. Please adhere to the safe management measures. Maintain a safe distance of 1 meter apart. Sit in the designated seats here in the LT, and please keep your masks on throughout. In the event of an emergency, please follow the directions of the ushers and evacuate in an orderly manner. We also kindly remind everyone to please put your phone to silent mode. At the end of the masterclass, please exit the lecture theatre using the door on the right of the screen. There will be a Q&A session at the end of the masterclass, but feel free to send in your questions via the QR code, which you can see here on screen. Or you can visit pollev.com/Q&A-LT27. That is pollev.com/Q&A-LT27. For those who are joining us via the webinar, in the event that the Zoom session ends or disconnects unexpectedly, please rejoin us at the same link. Our speaker for today is Associate Professor Christoph Winkler from the Department of Biological Sciences. Prof. Winkler's current research areas are developmental biology, neurogenetics, and molecular cell biology. In this masterclass, he will touch on how small fish are used to discover molecular pathways that allow bone cells to control bone cell activity, paving the way for new therapeutic strategies. Let's welcome Prof. Winkler.
Thank you very much, Janice, for the very kind introduction. Very good afternoon and a very warm welcome to this open house masterclass of the College of Humanities and Sciences. So my name is Christoph Winkler. I actually joined NUS 15 years ago in 2007. I'm originally from Germany. And I work at the Department of Biological Sciences. And what I'm going to do today is tell you a little bit about research or science that's at the transition zone between biology and medicine. Tell you a little bit about the research we are doing and others are doing using small laboratory fish species such as the zebrafish or the Japanese medaka to address problems in human disease biology, questions that have not been resolved on human diseases. So what you see on my next slide which is not here, is a zebrafish embryo at 16 hours post-fertilization. And this structure has actually kept me busy for the past 30 years. And I have been, and I'm still very much fascinated about the beauty of this structure and the fact that this beautiful structure forms in only 16 hours after fertilization. So from a one-cell embryo, from a one-cell egg, 16 hours later, you have a structure like this, where you can see the brain forming, an eye forming, the trunk and the tail forming, and you can also see patterns that form in this early embryo, right? Ultimately, of course, scientists want to understand how this structure forms. I don't know why. Okay, how this structure forms, which is the human brain. And just imagine the sheer number of neurons we have in our brains, over 100 billion neurons. And this is remarkable. And just imagine one step further, each single of these 100 billion neurons needs to be formed at the correct time, at the correct place, and it needs to make connections to its neighboring cells in a very precise manner. So that's a level of complexity that's sheer awesome, right? And it's very difficult to understand. It's difficult to assess this organ, of course, and it's also difficult to image individual cells in this organ. So we need model systems that allow us to look into the brain, maybe slightly more simple brain, and look at individual cells. And the zebrafish could actually be a promising tool to do that. So a group in the USA, Dr. Philip Keller, they actually advanced one step towards the goal of understanding the function of each individual neuron in a brain. In this case, this is a zebrafish brain, and you can actually see the brain here from the top, different compartments of the brains. There's an eye here and another eye here. This is the brain from the side, and this is a cross section. And what Dr. Keller managed to do is he genetically modified these fish so that each individual neuron will actually emit fluorescence when it's active. And then when I start the movie playing, you can appreciate how dynamic neuronal activity is in this structure. You can see individual neurons firing, getting activated. When Dr. Keller looked very carefully, he could actually see patterns. He could see that when one neuron gets activated, a neighboring neuron gets activated, and he could identify patterns in the structure. So in this particular case, the zebrafish larvae is anesthetized, it's sleeping, right? There's no challenge from the outside. But you can use a similar method now to look at brain activity in an environmentally more relevant situation. So in this case here, a group in Japan, the Kawakami lab, they used the same approach. They used a genetic trick to make neurons emit fluorescence when they are activated. And then they basically labeled neurons in a particular region of the brain and analyzed neuronal activity when this fish larva here was facing prey for food that the fish wants to eat. So when I start the video, and maybe you focus first down here, what you will be able to see is that as the paramecium here, this one single-celled prey, approaches, the fish sees the prey and then eats the prey, right? And now focus your attention when the video plays again onto the brain region. So these are close-ups views here. So if you look, for example, here, as the prey is approaching, very low neuronal activity, but after the food is eaten, you see very high neuronal activity. And actually that helped to identify neurons that control feeding behavior and that control satiety feeling in this fish larvae. When you take out these neurons, we can do this by genetic tricks, the fish will actually not stop eating, it will continue eating because it has no feeling for satiety. So fish are a wonderful system. One important feature is they are transparent. So we can look inside the fish larvae to look at cellular processes, and also development is very fast. When I play this time-lapse movie, you see the development from 30 minutes after fertilization up until 20 hours. Initially, there's just uniform cell division starting from the two-cell stage, then cells start moving, an embryonic axis forms, and after 20 hours, you can actually already see the head forming here with the different brain compartments, the eye, the trunk, and the tail, and these muscle segments here, right? And this all happens in the first 20 hours of their life. After 48 hours, these larvae are free swimming, they hunt for food, and then after 3 months, they become sexually active and they produce the next generation of embryos. So why do we use fish in science? It's actually a very popular model in research. The first reason is it's very easy to keep in the laboratory and in large numbers. So this is actually a view into our fish room. You can see we have these racks with fish tanks. Each tank can hold up to 50 fish. And it's actually very easy to keep these fish in the laboratory. It's easy to obtain embryos in large numbers. So what we usually do is the night before we plan an experiment, before we need embryos, we put a male and a female fish, zebrafish, into such a mating tank here. The next day, as soon as the light is switched on, mating starts, and you can see The male, in slightly dark, is hunting down the female, but no worries, the female also enjoys that. And then the female will release the eggs, and the male will fertilize the eggs. Fertilize the eggs. The eggs then will fall down here through a mesh and into the bottom of the container. It's important to separate the embryos from the adult fish because the adult fish cannot differentiate between food and embryos, right? So they would eat the eggs. Hence, we need this mesh in between. A female zebrafish produces approximately 200 embryos per day. So we have access to large numbers of embryos. They develop rapidly, as I've shown you. We know all the genes in the genome of these fish. They are very well identified and also characterized. We have a rich genetic toolbox. We can do transgenic fish to activate genes, or we can knock down or knock out genes. We can also label cells and do cell transplantations. We use fish for drug screening, and I will give you an example of that. But the most important point is because fish larvae are so transparent, we can use advanced microscopy to really look inside the embryo and look at dynamic cellular processes. And that's the big advantage to other animal models, such as the mouse, where this is not possible because they are not transparent enough. So it's no surprise that zebrafish, are very popular in the scientific community and are actually used to model many human diseases. There's hardly any human disease that cannot be modeled in the zebrafish system. The community is large, so worldwide we have more than 1,500 labs that use zebrafish as a model. In Singapore alone, 15 labs in the different university and research institutes. More than 10,000 PhDs work on this fish model. Today, I want to give you two examples. I want to briefly talk about a zebrafish model for a neurodegenerative disease, and then I'll talk about another fish, the medaka, Japanese medaka fish, as a model for a bone disease. So first, the neurodegenerative disease is actually a very devastating human disease called spinal muscular atrophy. And in this disease, what happens is that motor neurons, a particular type of neurons that controls muscle activity, degenerates and dies. And as a consequence, human patients suffer from a progressive paralysis and muscle wasting. Very often, this leads to premature death. In fact, 50% of the children that are diagnosed with spinal muscular atrophy, they die in the first 2 years of their life because they suffocate. The muscle that control breathing fail and they suffocate. Very devastating. So this is the most severe form of this disease, type 1. But there are also milder forms, type 2, 3, and 4, that have milder symptoms and a later onset. So this is how the doctor usually finds out initially whether a child has spinal muscular atrophy. When they pull at the arms or they hold the belly and there's no muscle tone, that could be a first indication that there's something wrong with muscle control, right? And then further analysis is necessary. It's a frequent disease. So 1 in 6,000 newborns actually suffers from the disease. And this makes it the most frequent genetic cause of early death. And the gene that's affected is actually known for a long time, more than 25 years. It's called survival motor neuron, or SMN. And here starts the puzzle or the enigma, because it's well known what this gene does. It encodes a protein that controls a housekeeping function that's necessary in all cells. For those of you who have a biology background, I'm sure you've heard about mRNA splicing. And this is exactly what SMN is doing. SMN controls mRNA splicing, the removal of introns, non-coding introns from an mRNA transcript, which is called mRNA splicing. This is SMN's functions. And it's doing this for thousands of genes in billions of cells. So the big question then is, why are only motor neurons affected? Although there's a deficiency throughout the body, selectively only the motor neurons get affected, die off, and lead to paralysis and muscle wasting. So to find out how SMN causes this disease, it's really like searching for the needle in a haystack. So what we did is we turned to zebrafish as a model. For the simple reason is because we can look inside the embryo. So these are labeled motor neurons, and you can see dynamic processes going on in these neurons. And this really allows us to go very deep, to look very deep inside and figure out what's going on and what's going wrong under spinal muscular atrophy conditions. So what we did was we actually looked at these motor neurons in the zebrafish where SMN, the disease gene, its function is impaired, and then looked at targets in the motor neurons that could be affected by a deficiency in SMN. And we identified one particular protein, which is called neurexin, which sits here at the terminal of a motor neuron. So the motor neuron comes in here, it makes contact to a muscle cell, and this structure here is called a synapse. And we found this protein that sits here at the terminal of this motor neuron and binds a partner on the opposite side of the muscle fiber. And what it turned out is when we mutated this protein, we got motor neuron defects. So this is the situation here in a control embryo. These motor neurons nicely formed, and in embryos that were deficient for this neurexin protein, you see all these defects in the motor neuron. So we have identified one of many needles in the haystack, and we were very happy to find that this protein is also affected in mouse models that suffer from spinal muscular atrophy. We found one other interesting feature by accident, coincidence, and that is that the mutant fish actually change their social behavior. So usually what happens if you put fish into a new tank is that initially they're very scared and they stay at the bottom here and show signs of anxiety. And this is actually very similar, whether this is a control wild type fish or the mutant. A difference then happens slightly later. In the wild type case, the fish become more brave, more curious, and they explore all places in the tank, right? The mutant, however, it's still very scared. It has still increased anxiety and affected social behavior. So this is something we are analyzing now, how a deficiency in neurexin actually affects behavior, anxiety behavior, and social behavior. I want to finish this part with a positive outlook. And I want to be very clear here. So this is about new drugs for treating spinal muscular atrophy. And I want to be clear, the zebrafish has not helped to identify the drugs, right? The credit here really goes to mouse labs, especially one lab in the United States, who identified a small drug that is able to rescue spinal muscular atrophy in a mouse model. So what you see here is actually a mouse that suffers from spinal muscular atrophy. And here it's this sister mouse that was treated with this drug. And it completely rescued the disease phenotype. This observation led to the conduction of a clinical trial, started in 2011 with a small number of subjects, of individuals. One of them is this girl over here. And remember, 50% of children diagnosed with the disease die at early age, right? So this girl here was unable to stand, to walk. She was bound to a wheelchair. After one injection of the drug, she was able to stand, and she's actually fine today, right? So very efficient and successful use of the drug. Because this was so successful, in an unbelievably short time, FDA gave approval for this drug. So only 5 years from the launch of the first clinical trial to FDA approval. Nowadays, we have two drugs that can treat spinal muscular atrophy very successfully, very efficiently. One is the drug that I just described, Spinraza, requires 6 or more injections. Look at the price. A single injection, $125,000 US. And there's a second drug that was approved in 2019. Good thing, you only need 1 injection, but look at the cost, US dollars 2.1 million. So these drugs are extraordinarily efficient, but they are very, very expensive. And that's a problem, of course, for our health system. How do we deal with this, right? So now I want to switch gears, and in the second part of my talk about bone and the bone disease, osteoporosis. And the first questions you might have is, you want to use fish as a model for a bone disease. Do fish have bones? Yes, fish have bones. You all know when you eat fish, we have a bone to pick, right? So fish have bone. The bony skeleton looks very different from a human skeleton, but actually bone, the bone cells and the genes that control bone formation are very similar, if not identical, between humans and fish. So our aim is actually to use fish to model the human bone disease osteoporosis, which mainly affects the elderly, which mainly predominantly affects females, women, and leads to bone fractures, an increased risk of bone fracture. If it happens here in the vertebral bodies, it leads to these twisted spines, right? So bone, you probably don't know, bone is a highly dynamic organ. It's constantly remodeled. As you sit there now, actually your bone is getting remodeled. Old bone matrix is resorbed by cells that are called osteoclasts. And this resorbed old bone is replaced by new bone, which is formed by another cell type called osteoblasts. And this is important. This constant remodeling is important to keep your bone healthy and rigid, right? So there's a balance between these bone resorbing cells and bone forming cells. And to achieve this balance, these two cell types, they need to communicate with each other. Some factors are known, like this rank L factors, others are unknown. There's a big question mark how this communication between these two cell types actually occurs. So under conditions when this balance here is impaired, for example, you have more bone resorbing cells, osteoclasts, than bone forming cells, problems occur. There's more bone resorption than bone formation, and that's actually why, or what leads to osteoporosis, reduced bone mineral density, and then an increased risk of bone fractures in the vertebral bodies, in the wrist, and in the hip. And this is really a huge health problem. If you look at people over the age of 65, 50% women and 20% of the men have an increased risk of bone fractures. So it's a huge burden also to our health systems. So we are interested in solving this question mark here, identifying new factors that are involved in this communication. And for this, we use the Japanese Medaka. Why are we not using zebrafish? Because Medaka from Japan forms bone earlier than zebrafish. It's just a very simple, practical reason. So here we want to identify novel genes that are involved in bone cell communication, and we want to do drug screening to identify new drugs. And why we use fish is nicely illustrated here. It's transparent. You can look inside into a living fish, and then after a genetic trick, you can see the bone forming cells in red and the bone resorbing cells in green. So it really allows you to study the dynamics of these cells during different processes. We established a Medaka model for osteoporosis. So we made a transgenic fish where the number of osteoclasts can be increased by a simple trick by just keeping the fish for a short time at slightly elevated temperatures. This leads to the production of this osteoclast-inducing factor rank L, which we can visualize by blue fluorescence. And then the consequence is that these fish produce excessive numbers of osteoclasts. So we copy this situation here. So at that point, there would not be many osteoclasts here in the vertebral column, like you see here. This is empty, but after rank L, there's a lot of these osteoclasts, and they are actively resorbing bone. When we look at the bone matrix here stained in red, in a control sibling, you see nicely formed bone in one vertebral body here in the center and in this spiny structure here. And then after induction of osteoporosis, we get a complete resorption of these spines, and we get large holes here in the vertebral body. So that's very typical hallmark of osteoporosis. If this is really osteoporosis, we should be able to treat it with drugs used in human osteoporosis therapy, right? Like, for example, Fosamax. That's one of the commonly used drugs. So I'll show you how we did this experiment. Unfortunately, because of COVID restrictions, I cannot bring you to the lab or to our fish room. So I thought I'd show you two short video clips, how we collect the embryos and how we do the treatment.
To harvest the embryos during the first few hours of the light cycle, first use a fine meshed net to catch a female adult carrying an egg cluster. Let the fish briefly rest in the net and then gently massage the fish's abdomen to carefully strip the fertilized egg cluster from the abdomen of the female. Transfer 20 to 30 eggs into individual plastic 60mm Petri dishes and use a plastic pipette to rinse the embryos in each plate with 5 to 10 milliliters of NUS solution. Gently roll egg cluster to form a knot of attachment filaments. Using forceps, carefully remove the attachment filaments from the fertilized egg clusters to obtain individual embryos. Then, culture the embryos in a 28 degrees Celsius incubator with daily medium changes to ensure normal embryonic development.
So this was Ting Sheng, a PhD student.
For live fluorescence imaging of the Medaka larvae, use a plastic microloader to orient the anesthetized larvae according to the region of interest, and place them under a stereo microscope with fluorescence illumination. Select the appropriate magnification and image individual sections of each larva using a suitable image processing software to stitch together the images at the appropriate overlapping regions. After each larva has been imaged, return the animal to its well in the 6-well plate in fresh fish medium for recovery.
Yeah, and that's the point then when we add the drug. So we don't inject the drug into the fish or anything. We just add the drug into these small wells into the medium, and then the fish takes up the drug, right? And what we found was that this is the control situation that I've shown you before without any transgenic induction of this osteoclast inducer. The vertebral bodies form nicely with this nice spine. When we add the osteoclast-inducing factor, many osteoclasts form and they resorb the bone, no more spines and these holes here in the vertebral bodies. And now what happens when we add the drug, Fosamax, osteoclasts still form, but they are no longer active. So now the bone almost looks like in the control. We see the spines and we see very little of these holes. So that means in our fish model, a human osteoporosis drug actually works. That tells us we can use this model to screen for drugs, and that the mechanisms of osteoporosis in fish and in humans are very similar. So we took advantage of this now and purified individual bone cells. I won't go into the detail because I'm running out of time, but basically we sort these fluorescent bone cells so that we can get individual cells. And then we do a genetic analysis. We sequence all the RNAs and we look for genes that are upregulated under osteoporotic conditions. And what we found was that a gene encoding a small protein called CXCL9L is strongly upregulated under osteoporotic conditions. So this protein here binds to a receptor protein that sits in the membrane of osteoclast progenitors or precursors and activates this receptor. And this is novel. This is the first factor that is known to activate osteoclasts and recruit them to bone matrix. So this is how it works. So here we have bone matrix, and under normal conditions, there are bone-forming cells here sitting on the surface. Under osteoporotic conditions, these osteoblasts here are instructed to produce and release CXCL9. So it will diffuse away, and it will bind to receptors that sit on the membrane of these osteoclast progenitors, which are actually macrophages. The binding here activates these macrophages, directs them, recruits them to the bone matrix, where they mature into osteoclasts and do bone resorption, right? So this is the first factor that is able to recruit osteoclasts to bone matrix. We know that these macrophages also secrete other factors that work on bone-forming cell progenitors and also recruits them to the bone to actually repair the osteoporotic lesion. This is something we are analyzing now. So nice to know that this works in fish, but ultimately, of course, we want to translate this into the human system. Does this have any clinical relevance? It does, because actually there are already 2 drugs available that inhibit the receptor for CXCL9L, for the small protein I was talking about. So these 2 drugs were developed by Amgen, and they were actually clinically tested in a phase 2 clinical trial, but not for osteoporosis. Amgen tested them for another disease, psoriasis, which is actually has nothing to do with bone, but it's an inflammatory skin disease. The drug went into phase 2, meaning it's safe for the patients, but unfortunately, it is not effective for psoriasis. So the clinical trial was actually stopped. So we took these 2 drugs and put it onto our fish model, and we found that treating our osteoporotic fish with these drugs protects the fish from osteoporosis, from osteoporotic insult. So this is the control, no drug added with the osteoporotic lesions, and this is after the drug was added. So we actually filed a patent, an international patent, because what we now want to do is bring this drug into the clinics and test it in osteoporosis patients. Okay, I end with the last experiment. Of course, we can use drugs to treat osteoporosis, but maybe there's an even easier way to prevent osteoporosis. And one approach could be look at your diet and use supplements in your diet. So there have been rumors around that a particular group of plant pigments has positive effects on bone cells. These pigments are called anthocyanidins or anthocyanins. And there's 2 particular chemical compounds, delphinidine and delphinidine glucoside, in this group that are found in especially plants and veggies and fruits with purple color, right? So grapes, blueberries, blackberries, but also eggplant. So these are rich in these compounds. So we wanted to test whether these compounds work to prevent osteoporosis. We use our osteoporosis model. After Rangel induction, we see all these osteoclasts, and then we added increasing concentrations of these plant pigments. And what we found was that in a dose-dependent manner, osteoclast formation or the presence of osteoclasts was repressed. Right? So by simply adding a plant pigment into the dietary mix, what we can do is reduce the number of osteoclasts. And actually, it's less osteoclasts that form in the presence of these delphinidine. But what delphinidine also does, as you can see in this video, it reduces the stability of these bone-resorbing cells, of these osteoclasts, and makes them die, right? So I want to end with 2 pieces of advice. Eat your berries. If you want to prevent osteoporosis, I think it's a very efficient way to put berries into your diet, especially black and blueberries, and keep the levels high. And my second advice to you is join us at the College of Humanities and Science and the Life Science program, because we do cool research. We offer undergrad research opportunities. You can come to our lab and do research projects. This is my lab. And every year we have between 2 up to 6 undergrad students who do research projects in our lab. So I thank you for your attention, and I'm happy to take any questions. Thank you.
Thank you, Prof. Winkler, for guiding us through this masterclass. We will now be opening the floor to questions. You may send in your questions by scanning the QR code here on screen or visit polev.com/Q&A-LT27.
And you can ask questions about the research, but also more general about the life science program, modules that we offer, biomedical. So I'm a member of our biomedical program. We have 3 programs in the department: molecular cell biology, environmental sciences, and biomedical sciences. I'm a member of the biomedical sciences, right? So if you have any questions, if you are biomedically inclined, then please let me know. What is the percentage number of students who have another minor or major while studying life? That's a very difficult question for me because I don't really know the numbers. But, and also, as you know, the CHS program is relatively new. Can I ask you to please contact our admin office? They will be able to give you the exact numbers. What are the career prospects of life science graduates? We addressed these questions extensively in the session that was aired on Monday, and I think you can watch the video again. The career prospects are manifold, just to give you a brief answer, right? I mean, many of our undergrad students after graduation, they stay in research or they join pharmaceutical company. It depends a little bit on the direction, of course, or they join government offices, especially when it comes to environmental sciences. There are many opportunities. I'm not the perfect person to answer this question, but on the open house session that was aired on Monday, our deputy head, Henry Mock, addressed this question in great detail. So maybe you can look at that video again. Is it true that life science graduates are oversaturated? What do you mean? If the question is they don't find jobs, then the answer definitely is no, right? The career prospects for life science graduates is very good. The job market is not oversaturated. If it means students are overloaded with knowledge, I don't know. Some students take more knowledge than others. Do students choose their own final year projects? The answer is yes. So basically you approach profs in the department. You either know through the models you take or you go to the websites of the individual labs. You have a particular interest in a particular research direction, right? You identify the prof that works in this area. You send him or her an email and you ask for available projects. But it's the student who chooses which PI, which prof to approach and which project to take. Is it highly competitive to do research during my undergrad studies? I mean, I can only tell you from my own experience. When I was an undergrad student, I spent most of my time actually in the lab, because to me, that is the fascinating part of science, is doing experiments, right? I spent clearly more than 50% of my time in labs doing undergrad research projects rather than attending classes. So of course it's time consuming. You have to plan your time wisely, but and it depends on your own preference, right? But if you like to do experiments, if you like to work in the lab, I definitely would try to take the opportunities that we offer with these undergrad projects, approach PIs, ask for projects, join the lab, and then get mentored by graduate students in the lab. You will learn so much, and you will see, actually you will see whether this is fun to you or not, whether you want to continue a career in academia or not, right? I strongly recommend. I have an interest in biomedical sciences, specialization. May I know what careers I can choose? Again, manifold, right? I mean, many of the undergrad students that were in my lab, they continued either as a research assistant or as a graduate student. They did PhD. But I also have undergrad and postgrad students who, after graduation, joined pharmaceutical companies, right? That's also possible. Actually, there's no one answer. There are so many opportunities. You just have to take them. What is something that most students struggle with in this course? There's no student here, right? I mean, of course, the class load, the load is relatively high. And the further you go, you know, you enter year 3, year 4, there's a lot to learn, there's a lot to do, and especially if you have a research project on the side, you need to really work on your time management skills, right? It's not easy. I'm not saying it's easy, but there's a lot of commitments, there's a lot of assessments to do, commitments that you have, right? So you need a good time management. And I think probably that's what students struggle most, to juggle all these different responsibilities. Is it difficult to continue to take graduate med or dentistry? So we definitely have students that after graduation from the life science program enter Duke NUS Medical School. Again, very competitive. Obviously, there's many candidates. You need to be very good, but it's an option. We have graduates that enter medical school afterwards. How's the pay like? My pay? I will not reveal. The pay, what students are not paid as far as I know. After graduation, it depends on the job. And again, I'm not a good person to answer this question. Will you be allowed to work on specific diseases of your choice? Is that Alzheimer's? Yes. Will you have to put in a request to work on certain research projects and get them approved before working on them? So you need to find a lab, a professor that works on that particular disease, right? Or any other project. And of course, your undergrad research project or your final year project is mentored by the PI, by the prof. So you cannot just go to a lab and say, I want to do this and start doing this. This will be under the supervision of a PI. You can convince the PI that this is an interesting project, which I think is a bit difficult. But if the PI is already working on Alzheimer's disease, and because this disease is mentioned, there are many labs at NUS that work on Alzheimer's disease. There's plenty of opportunities, right? Then you just write an email to the prof. You would be interested in a Europe's project or in an FIP project. Are there any projects available? And then if there's space, the prof will get back to you. For the experiments live fish were used, how do you ensure that the experiments are ethical? NUS has a very high standard of ethical regulations, right? So every single experiment that we do must be approved by our ethical committee, animal welfare committee. It's a long process. Actually, before you start the experiment, you have to write a proposal, submit the proposal to the animal welfare committee and get approval for this experiment. It's highly and tightly regulated.
Due to the interest of time, Maybe we'll take one last question.
Are there opportunities for overseas exchanges? Yes, absolutely. I mean, not in the past 2 years, obviously, but before COVID, our undergrad students extensively traveled overseas and did coursework or even research projects overseas. And even for your FYP, depends on your supervisor, on your prof. If the prof has a collaboration, he can send you overseas so that you work for whatever months in the collaborators lab overseas. So before COVID, I can tell you the vast majority of our undergrad students at some point went overseas for different periods of time.
Okay, thank you everyone for your questions. Thank you for all your curiosity. And thank you, Prof. Winkler, for answering our participants' questions. Should you have any other questions, you may send us an email at the email address shown here on the screen. We hope that you have been able to learn more about the course and enjoyed your glimpse into learning at today's Experience NUS Open House. There are many activities that have been planned for you today around the campus, such as student life activities happening at UTown. We wish you a good day ahead, and please exit the lecture theatre via the door here on my left. Thank you.